Hi guys. So this session, we are looking at the plasma membrane. I'll get myself out of the way and we can move on. Okay, so plasma membrane, I want you to kind of review the idea of phospholipids. Remember, we have our hydrophilic and our hydrophobic areas. Make sure you remember what's what. Okay. So A is our phosphate group. B is the glycerol. And the C is the hydrophilic head. Those pieces are water loving. And then D are our hydrophobic tails made out of fatty acids. Okay, so the plasma membrane separates the internal cell environment from the external environment. And you'll notice that it is a double layer membrane. So it's comprised mostly of phospholipids. And notice how they arrange themselves to protect themselves from the water. So they are what we call amphipathetic. Hydrophilic heads, hydrophobic tails. They're opposite each other. And so they naturally form a bilayer. This makes them selectively permeable so they can actually regulate what moves into or out of the membrane. So the hydrophilic heads always orient towards the aqueous environment because they like the water, and the tails are always facing inward away from that aqueous or water-based environment. And it does this naturally. So we call this a fluid mosaic model. That's kind of the common way we describe the structure of the membrane. It's fluid because we have this weak hydrophobic interaction, but they can still definitely move and shift. That's why the phospholipids are such an important part. Temperature can affect this, so it's going to flow more easily as it gets warmer, and it'll be harder for it to flow as it gets colder. The unsaturated hydrocarbon tails also help maintain that fluidity even at the lower temperatures, but they aren't going to flow as well. And the kinky tails, I know the kinks, those kinks, remember the kinks are from the double bonds in that fatty acid tail. They actually keep it from packing too tightly. It helps space them out. So cholesterol, I know everybody's like, oh, cholesterol, it's so horrible, but cholesterol is really actually very important. That's this yellow part. At higher temperatures, it kind of reduces the movement so it can't separate too much. And at lower temperatures, it keeps those phospholipids from packing too tightly together. So it's really important in terms of the structural component. And that's that yellow piece that we're seeing kind of there in the middle. So we call it a mosaic because it's made up of all these different types of macromolecules. So it's not just one specific type of molecule. So when we're talking about those proteins, there are two key categories for the proteins. We have what we call the integral proteins. They go all the way through. They're embedded into the lipid bilayer, go all the way through. Sometimes we also refer to them as transmembrane proteins or amphipathetic proteins. And then we have the peripheral proteins that you're going to find, they're really not embedded all the way through the layer. They're just kind of on the top or on the bottom. And so they're just loosely bound to the surface. The membrane-bound carbohydrates, like our glycolipids, so carbohydrate with a lipid chain on it, and glycoproteins, same concept. These are carbohydrates bound to lipids. So those are the green chains you see here. And glycoproteins are the blue chains bonded to proteins. We find more glycoproteins than we do glycolipids. But this is what they use to kind of mark and recognize each other. So these are kind of almost like their names, except they're a physical marking. Think about how we use Braille on things. So somebody, even if they can't see, they can still feel and go, okay, this is such and such. It's that same concept. So with plants, we have a cell wall covering that plasma membrane. So remember, that gives them a kind of a more formal structure. That extracellular structure is not found in animal cells, so we do not have cell walls. It gives shape and structure. It also provides protection for the plant, and it helps regulate water intake. It's made up mostly of cellulose, which of course we can't digest. This makes it a lot thicker than the plasma membrane, and it also contains something called a plasmodesmata. And the plasmodesmata are almost like little holes. So they can kind of connect cell to cell. So the cells can actually communicate with each other somewhat through the plasmodesmata. Okay, so the Chenoichthydae fish, also known as the crocodile icefish, lives in the Southern Ocean around Antarctica. We know that these waters are really low temperatures all year. They range from about 28 degrees Fahrenheit to 35 degrees Fahrenheit. So these are really cold. These fish actually have adapted to allow them to live in those cold, low oxygen waters. So if you were to examine the membrane lipid composition of cells from this fish, identify what you would expect to find that helps them live in these environments. And then you want to justify why you might find that part you identified. You're going to tell me why you think that. Okay, so hopefully that gave you a little bit about the membrane. Bye.